Gregor Mendel is widely considered to be the father of modern genetics. He studied the inheritance of traits in pea plants that were growing in his monastery's garden. He kept records and data on things like pod shape, pod color, seed shape, stem height, and so on. Mendel chose pea plants because they could reproduce quickly, and he could also control how they fertilize each other. He would take the pollen from one plant and transfer it to the pistil of a different plant. So this was a purebred cross, and this was known as the parent generation or the pea generation. So he would take the purple flowers and the white flowers and cross them, and that would create the F1 generation. And the F1 generation had only purple flowers, which was a little bit odd to Gregor Mendel, because, in the P generation, you're starting with purple and white flowers, and then in the F1 generation, the white flowers seemingly disappear. So he would then take the F1 generation and he would allow them to self-pollinate, and that would create the F2 generation. So in F2, he noticed that some of the white flowers would reappear in the offspring. So he looked back and he was like, okay, so F1, there were no white flowers. So it's almost as if that trait skipped a generation. Not only did Mendel observe things like flower color, but he went back and he looked at traits like pea shape, pea color, pod color, flower position, and plant height. And he actually counted the number of offspring with each trait, and he would assign one of the traits, the one that appeared the most, the dominant trait. He would call that the dominant trait. The other one that appeared in lesser numbers or in fewer numbers, he would call that the recessive trait. And he actually counted every single one. He would make a tally. And what he noticed was that there was always a magic 3-to-1 ratio for dominant versus recessive traits. So if you look at just pea shape, under dominant, he counted there were 5,474 peas that were round. And he also noticed that there were 1,850 that were wrinkled. So that gave him a ratio of about 2.96 to 1, which you can round to 3-to-1. And this was consistent throughout the other traits. There was always this 3-to-1, this magic 3-to-1 ratio. Mendel's studies have greatly influenced our understanding of modern genetics. Nowadays, we understand that a gene is a piece of DNA that instructs the cell to make protein, which then expresses a trait. Genes can have many different versions, which is why you can have traits like purple flowers or white flowers, or round peas versus wrinkled peas. You have 2 alleles for each gene. So for example, if you're looking at wrinkled versus round plants, if you have a homozygous set of alleles, that means you're carrying 2 of the same traits. So that means you would be carrying a wrinkled and a wrinkled allele. If you are heterozygous for a certain trait, that means you're carrying an allele for both wrinkled and round. So just to clarify, you can understand quite a bit just by looking at the Greek roots. Homo means the same, hetero means different. So if you are homozygous, that means you're carrying 2 of the same alleles, and if you're heterozygous, that means you're carrying different alleles. Now that you know the basics of genetics, we can actually look at monohybrid crosses and use something called Punnett squares to draw and predict the ratio of traits that will appear in the offspring. So if you look at the image to your left, notice how you're crossing a purple flower with a white flower, but 100% of the offspring are purple. Now if you look at the image to your right, you're also crossing a purple flower with a white flower, but now you're looking at half of the offspring being white. So why does this happen? We're actually now going to look at Punnett squares. I'm going to run you through a few examples, and you'll learn about homozygous and heterozygous crosses. Before I start showing work for these Punnett squares, you just have to understand a few technical things. When it comes to dominant traits, you can think of them as common or the superior trait. So dominant traits will appear quite often. On the other hand, recessive traits means that they're rare and they actually need 2 copies of those alleles to actually be expressed. So long as the organism has at least 1 copy of the dominant allele, the dominant trait will be expressed, whether that organism is homozygous or heterozygous for that trait or that gene. On the other hand, recessive traits or the rare trait will only appear if the organism is homozygous for those alleles. That organism will need 2 copies of the recessive allele for that trait to be expressed. When you do a Punnett square, you will assign the dominant trait with any capital letter. So in this case, purple is the dominant trait, so we will assign purple with the capital letter F. As for the recessive trait, you will assign it with the same letter, but the lowercase version. So in this case, the white flowers are the recessive traits, so you will assign white with the lowercase letter F. So let's clarify all of these different combinations that you can have for your genes. If you're homozygous dominant, you would have 2 capital letters. So for example, big F, and phenotypically, you would appear purple in color because purple is the dominant trait. If you're heterozygous dominant, you would have a copy of each allele. So in this example, you would have an uppercase letter and a lowercase letter, so a big F, little f, but phenotypically, you still appear purple. Even though you are purple as a heterozygote, you are actually a carrier for the recessive gene because you have a copy of that little f. It's just phenotypically you appear purple because purple is the dominant trait. And finally, you have homozygous recessive, which means you have 2 copies of the recessive allele. So in this case, you have little f, giving you a white flower. So let's do our first Punnett square, and in this example, we are taking a purebred purple flower, which means it is homozygous dominant, so capital F, big F, and this parent would appear purple in color. And we're going to cross this with a purebred white flower parent. So this parent would be homozygous recessive. So along the top, we want to place the first parent, so big F, and remember, this parent would appear purple in color. It would have purple flowers. And the other parent would be little f. It's homozygous recessive, which means it would appear white in color. It would have white flowers. And now this is how you do a Punnett cross. So in each of the 4 quadrants where the letters intersect, you just want to line up the letters. Okay, and so big F, little f goes in that box. In this quadrant, a big F followed by a little f. You always want to put the capital letters first because those are the dominant traits. In this third quadrant, big F, little f, and in the 4th quadrant, big F, little f. So it's just where the guys intersect, where the letters intersect. Okay, so now this offspring here, and this one here, and this guy here, and this guy here in the 4th box, they would all appear purple. Because remember, the recessive allele is actually hidden. You need 2 copies of them for that recessive trait to be expressed. So in this case, all the offspring are heterozygous, dominant, and they would all have purple flowers. Okay, so it's like the white flowers just disappear in this generation. So one thing to note is the genotypic ratio is what we actually see in the boxes. Like, what are the genes of the offspring? And it turns out 100% of them are big F, little f, so they are all heterozygous, dominant for the purple trait. And phenotypically, so the phenotype is what you actually see. Okay, so the phenotype is the trait being expressed, and they are all 100% purple. So that's what a Punnett square could tell you. It could tell you what the flowers look like, and then in this case, we actually know that there will be no white flowers in the offspring of this generation here. Even though we started with a white parent, it's almost as if that trait disappeared because remember, it's a recessive trait, which means you need 2 copies of it to be expressed. If you look at all the offspring, which I'm circling right now, they're not homozygous recessive. They are heterozygous dominant for the purple flowers. So let's see what Mendel has done so far with this Punnett square. He has taken purebred purple flowers and purebred white flowers, and he has crossed them. So that was the parent generation, and the offspring, known as the F1 or first filial generation, all had purple flowers. 100% of them all had purple flowers. But keep in mind, the F1 generation, those guys are heterozygous dominant. So now we're going to cross those guys. We're going to allow them to self-pollinate, and we're going to see what's going to happen in the next generation. So let's see the next step of Mendel's experiment. We left off with the F1 generation, which were all heterozygous for the purple flower trait. So big F, little f. Now I just want to point out that neither of the parents in this case have white flowers, because in order to have white flowers, you need to be homozygous recessive. So that was not the case in this part of Mendel's experiment. So we're starting with both parents having purple flowers and being heterozygous for the trait. So that means they were both big F, little f. When you carry out the cross in the Punnett square, you just want to make sure you're writing down the correct letters, whether they're uppercase or lowercase. So where they intersect, that's what you actually write in the box. So in this box here, it's big F, big F. And you always want to write the capital letters first, because capital letters are the dominant trait, and the dominant trait takes over. That's the one that is expressed. So in this box here, big F, little f. So far so good. But down here, there is an anomaly. So you have little f, and Mendel will actually take note of this, and I'll explain in a little bit. Okay, so now let's figure out what the colors of the flowers might be. So big F means you're homozygous dominant, so you would have a purple flower. Big F, little f is also purple, so you're heterozygous dominant in this case. This guy is also big F, little f, so you would have a purple flower. Now I'm going to circle this one because little f means that you are homozygous recessive. So you would actually express the white flower trait. So a few things to do. Let's write down the genotypic ratio. So the genotypic ratio would be 1 big F. Okay, so that's 1 homozygous dominant. And we're going to see 2 heterozygous dominant, and I'll circle them. So it's big F, little f, big F, little f. So there's 2 of those guys. 2 heterozygous dominants. And finally, you have the homozygous recessive little f, little f. So just one of those. So the genotypic ratio is 1 to 2 to 1. And the last thing to put down is the phenotypic ratio. And the phenotypic ratio, again, is just what appears in your traits. Okay, what colors do we see? So I see 3 purple flower traits in the offspring. So 3 purple flowers. And again, this is just a ratio. 3 purple for every 1 white flower. Okay, so 75% of the offspring will be purple. The other 25% will be white. Okay, and it's interesting to note that even though your parents, the parents that we started with, had purple flowers, okay, both of the parents had purple flowers, it actually gave a 25% ratio of offspring that actually had white flowers. And that was really interesting for Mendel to note, because it's almost as if the white trait reappeared in this generation. Mendel's observations, as demonstrated by these Punnett squares, was groundbreaking. So what he noticed was the white flower trait actually skipped over a generation. It appeared in the parent generation, it skipped over the F1 generation, but then it reappears in a 25% ratio in the F2 generation. So you might be wondering to yourself, how can 2 purple flowers cross and create offspring that have white flowers? Well, if you think about it, if you look at the picture of this family, neither of the parents have red hair, but their child has red hair. So that means these parents are carriers for the red hair gene, and this child happened to get 2 recessive alleles, and that's why she has red hair. I think by now you have a much better understanding of dominant and recessive traits after having done the Punnett square crosses with me. In the next video of this series, I'm going to go over why certain human traits are more common than others. So for example, how come brown eye color is more common in human populations than blue eye color? We'll look at pedigree analysis, we'll do more dihybrid and monohybrid crosses, and we'll start to understand genetics more in depth. Thanks for tuning in to my lesson, and I'll see you next time on Win Biology.